go ahead and get started. It's now 10:02, so you might hear some beeps and that kind of stuff. I'll have to admit people who are waiting in the lobby trying to join. So I might be a little bit interrupted here at the beginning, but we're going to run this the same way I did my last Q&A on our virtual environment. I hope you are all doing well. It's nice to see you, although all I see is just your initials. So, but anyway, it's nice to interact with you, at least in a live environment. So this has been truly a strange semester. I'm sure all of you recognize, in a strange time of our life. In fact, I was in Old Town last night. It was my son's birthday, so we decided to go and pick up some food, and it's so dead there. It's crazy. So anyway, let's talk first about just kind of structure of the exam again. So the exam is going to be the exact same structure as last time that we did it virtually, so there's no real changes there. The exam is over chapters 12 through 14, so it's not cumulative, at least not in the strict sense. Of course, this class is, as you hopefully all recognize, has built upon itself. So you are going to have to be familiar, of course, with terms and concepts from chapters 1 through, I guess it would be 11, but I'm not going to test you directly on them. I think it'd probably be a decent idea to go ahead and review glycolysis and gluconeogenesis, because those concepts continue to come up because they're so intimately connected with, of course, the topics in chapters 12 through 14. So keep those things in mind. And also, in terms of the exam, it's going to be open starting at 8 a.m. And it's going to go till 6 p.m. on Monday the 11th. So you'll have 90 minutes to take it. The exam has been written so that you could take it in 50 minutes in an in-class setting, but you'll have 90 minutes to take it. So I think that everyone should be able to get done. The average time it took for people to do the last exam was only 50 minutes. So, or I think right at 60 minutes. So anyway, so I don't anticipate 90 minutes being a problem for anyone to get this done. So, anyway, so that's the general idea of the exam. For the Q&A, what you see hopefully on your screen is the Poll Everywhere exam for Q&A. People are already populating with questions, so we can start working on that. I did want to address real quickly, while I'm addressing, just kind of details here. There is this question: will grades be fully updated before the test? So I'm going to at least try and get those quiz grades up. I don't think I'll be able to get the extra credit up, at least not all of it anyway. I'm pretty busy right now, as you might expect. So trying to crunch the numbers on terms of extra credit and that kind of stuff probably won't happen, at least not by the time you've taken the exam. So, but I'll try and get it as updated as I can within a reasonable amount of time. Okay. All right, so let's go ahead and start answering questions here. Of course, feel free, coming back to our Microsoft Teams environment, feel free to go ahead and unmute yourself and ask any question you want. I do ask that right now that you go ahead and mute your mics if you don't have anything to say. That way we're not hearing your dogs bark or whatever, or your cat's meow or whatever, other animal you have, or your roommate do whatever they're doing. So anyway, but feel free to interject verbally. I don't mind certainly that. I'll go ahead and put up the chat here. I think that here's the chat. So you could type a message there as well. So and I'll run most of it though off of this Poll Everywhere stuff. So okay. So can you explain the lipoate arm? Let's go ahead and try and explain that arm. So this is going back to chapter 12 material, and the lipoate arm is essentially this part of E2 that can reach into the other parts of the PDC complex. So let me go ahead and try and diagram this for us. So hopefully there's not too bad of a lag time on your end as I diagram this, but let's just do this. Try and represent the PDC, pyruvate dehydrogenase complex, is just 3 essential shapes here. Okay, and we'll put one there, we'll put one right here, and then we'll go ahead and put one down here. Okay, and so we'll call this one E1, and we'll call this one E2, and we'll call this one E3. Okay, I just got beeped at. Let me real quick go back here. I think that, oh, okay, I thought that someone might need it in, but okay, anyway, coming back here. So E1, E2, and E3. So that arm that is being referred to, okay, is essentially found in this E2 domain, and it can reach into, okay, these different complexes, including itself, and kind of swings around between the 3 different, not complexes, but enzymes or subunits of the complex. And so what you're seeing here is the arm in not 3 different arms, but the arm in 3 different positions. Okay, and this is hopefully drawing back on you know, the figure that you saw that we used to describe the 5 steps of the PDC. In that version, there's not different arms being represented. It's just the arms in different position, and the arm can actually quite literally move between these complexes. Okay, and pick up things. Okay, and so in regards to E1, what it actually will do is it will, E1, if you remember, is going to decarboxylate pyruvate, and the arm is going to pick up the decarboxylated pyruvate, which is just an acetyl group. And then it's going to bring that acetyl group over to E2, where it will make acetyl-CoA, and then it will take electrons to E3 and give those electrons to FAD, and those will give it to NADH. So let me do this, because I don't want to try and draw all that. Let me just go ahead and reference you back, because my drawing skills just get, you know, I mean, they're really bad. But let's just do this, see if I can't pull this up for people here. Okay. Try and pull up the figure that I'm referring to here. Do that. Maybe. There we go. Oh, it pops up to the one that I've already have here. So, so this is essentially what I was drawing on my last little slide there, but this is in a lot more detail. Okay, and so what we have then, and again, is this arm. There's lots of writing already on this. So let me go ahead and erase some of this for you. Okay, so we can get rid of some of this detail here. There we go. Okay, but this arm here is this guy. Okay, that's the arm, and that's in its is one of its positions, and then it's going to swing through here, and it's going to take these carbons off of this TPP molecule that originally came from pyruvate. And those carbons are going to be attached to the arm as it moves through here. And so this is the second position of that arm. Okay, same arm as what we saw down here, but now it's in its another position. And then it's going to swing through its own active site as we move through this direction here, and it's going to make acetyl-CoA. Well, these carbons here are the same carbons that are shown over here, right here, where I just put that star. Okay, and so we've got then the production of acetyl-CoA, and then the arm. So we'll move again, and it will come here, and this is another position of the arm, looking down here where I just put that red star. Okay, and that is the reduced form of the arm that will then take electrons and give them initially to FAD here. Okay, and then those electrons are going to ultimately end up on NADH. Okay, so that's the arm. It carries both carbons and electrons in the pyruvate dehydrogenase complex. Okay, and hopefully that clarifies for you what the lipoate arm is. Okay. Okay. So moving onward, if there's not any more questions about that, if there are, go ahead and interrupt me. Again, use your mic or go to the chat. Let me make sure that we don't have something happening in the chat here. So, okay, great. Okay, so let's go back to here, our poll everywhere question, and let's look at the practice short answer or practice exam short answer. So I pulled this up on another computer screen, so I'll go ahead and read it to you. I figured some of you would ask about this. So it says this, in his study of alcoholic fermentation by yeast, Louis Pasteur noted that the sudden addition of oxygen to previously anaerobic culture of fermentating grape juice resulted in a dramatic decrease in the rate of glucose consumption. This effect has been called the Pasteur effect and can be counteracted by the addition of 2,4-dinitrophenol, an uncoupler of oxidative phosphorylation. A, why would these yeast cells consume less glucose in the presence of oxygen? And B, why would DNP counteract or prevent the Pasteur effect? Okay. Okay. Please say you're wearing the vest. I get a red check minus today. I'm just not wearing it. I thought about it. I even thought about it. I didn't realize I was going to get a red check minus. Now I'm really upset at myself. I did, and the last one I had to go even put it on. You know, here's the thing. I think that this whole COVID-19 and quarantine thing has just made all of us, you know, an entire generation of us return to bare feet. You know, I mean, we all just kind of sit around in front of computer screens. And so I'm not wearing the vest. I'm not in my outfit. But anyway, don't worry. The world is a stable place. Okay. But anyway, but back to the practice exam, short answer on the practice exam. So let's go ahead and look at this and talk about this conceptually. I'll come back to this here. There we go. Okay, I'll leave this and go ahead and think about the effects of this. And so this is, I think, a really great opportunity to kind of make some connections here, broad scale connections. So let's do this. Let's start here. Okay, and we will start with glucose. Okay, we could do the same thing for fatty acids. That would be awesome too. And maybe we will, because it would be fun. Okay, but we'll do glucose being converted to 2-pyruvate. Okay, and of course that's going to make a whole bunch of NADH. Not a whole bunch of NADH. It'll make certainly some NADH. Okay, so let's go ahead and do that. NAD plus, going to NADH, to those, and of course down here we'll just write out the net 2 ATP is going to be made in this process. Okay. Now, what we have learned, of course, now, and we have seen in a lot of detail in these last several weeks and in these last several chapters, is how pyruvate then can move into this environment. There's a transporter that moves pyruvate directly through these membranes. Okay, so we can move pyruvate in here, and pyruvate can then be converted, as we know, by the PDC complex to acetyl-CoA. And that makes some NADH. Oops, there we go. NADH. Okay, and then acetyl-CoA, as we know, can then enter into yours and my favorite cycle. Okay, the citric acid cycle. Become citrate, and then we'll just draw, another arrow connecting that and saying that that's the cycle. And of course, the outcomes of this, we get I'm going to put ATP. It's GTP, or it could be ATP. It just depends on tissue type and isoforms of enzymes, but we'll just put ATP. Okay, there's a small amount of it that's made. Okay, and then we also, of course, get from this NADH, quite a bit of it. There's 3 of them here that's made. Okay, actually 6, because I'm doing this per glucose molecule. Okay, and there would be 2 FADH2 made from this as well. Okay, and so those are the products of the citric acid cycle. And I guess I could say that, you know, we've got 2 CO2 here, and we've got the 2 CO2 here. Okay, not 2, I'm sorry, 4 CO2 there. Okay, now that then, of course, all connects to this wonderful, super awesome, I think that in one of my Canvas posts, I called this the mostis radist process known to man. So, you know, if you're reading those things, I'm sure that you're rolling your eyes right now and thinking, what a dork. But anyway, that's okay. I prefer geek, but dork is okay as well. It's just not what I have my fantasy baseball team named, which right now is a depressing subject for me, so we're going to leave that. And anyway, okay, but my fantasy baseball team on paper looks really good. But anyway, so these NADH, right, and these FADH2, and that would be true of these as well. And of course, we have these, and they have some shuttle mechanism to get in there. But we'll just kind of say that these are all going to feed into this process, and they're going to make NAD+. I'll kind of just ignore stoichiometry from this point forward. Okay, and of course, you're going to have oxygen. This is the really important part of this. Oxygen is going to be converted to water in this, and we're going to get this massive movement of protons through this process out to here. Okay, and then we've got the mostis radist enzyme known to mankind, right? Again, objective statement, right? So there's ATP synthase, and that is like the leastest, raddest depiction of that enzyme, but you get the idea, okay? And those protons are going to flow through here, okay, and they're going to go round and round they go, and out they come, and of course that's going to make a large amount of ATP, okay? All right, so lots of ATP can be made from that, okay? And so we've got all of this going on, and as long as you have oxygen, then you're going to make, you know, depending on the number of subunits in your C ring, you're going to make somewhere between 30 to 32 ATP per glucose molecule, okay? Now, when you go anaerobic, okay, you're removing this, okay, which means that you're not getting this ATP, and you're not getting this process, and you're not getting this, and you're not getting this, okay, and you're not moving pyruvate into here, okay, and you know, so there's you just lose all that ATP production that's happening in the mitochondria, and really when you're lacking oxygen, the only ATP you can make is from glucose. Okay, conversion to pyruvate, and in yeast, because that's our short answer question, references yeast, the pyruvate will be converted to alcohol, okay, actually ethanol, to recycle the NAD+. All right, but you'd have to then, in that case, consume quite a bit of glucose to make or meet cell demand, okay, and so that's right. That's what's happening in the anaerobic environment. Okay, now you're only getting 2 ATP per glucose. So going back to our question, it says, why would the yeast cells consume less oxygen in the presence of, or less glucose in the presence of oxygen? Well, the reason is because once you add oxygen to this whole thing, you get all of this back. Okay, which means that glucose can be consumed and make a lot more ATP per glucose molecule, and so you have to consume less glucose to keep up with energy demand. Okay, so that's why they would consume less oxygen, or sorry, less glucose in the presence of oxygen. Okay, all right. Now, why would DMP counteract that or prevent that? Let me check here. I thought I heard someone need it in. Okay, no. Okay, great. So now why would DMP counteract that? Well, DMP, so let's come here and let's go a little bit more focused on our mitochondrial membrane here. Okay. And I'll get a little bit more detailed with my picture here. So now I'll put ATP synthase, use similar coloring scheme that I was last time. Okay, still bad drawing. Okay, but you get the idea. And then you get the electron transport chain over here. Okay, that's taking the NADH. And it would take FAD too. Okay, we're just ignoring that for right now, but it would do that. And converting the oxygen to water and moving those protons, like we said, massive amounts of them, to the side, and then those protons go back through here, okay, around they go, and of course we get the ATP production. Okay, right. Now, DMP, so why would it counteract this? Well, going back to Louis Pasteur's experiment, right? So we have essentially glucose making 2 ATP per glucose molecule without oxygen. You add oxygen, and you can get about 30 to 32 ATP per glucose molecule. So you get 16 times more ATP per glucose molecule, so the cells stop using as much glucose. And then you add DMP back, and all of a sudden they start to consume more glucose. Well, why would they consume more glucose? Well, the reason is because DMP does this. DMP grabs onto this. Some of these protons will be attached to DMP. That will make this DMP. And we'll put a pink proton on this. Okay, those will move through this membrane. DMP will come here. Forgot the N. Plus proton. And now what we're doing is we're essentially moving protons across the membrane and not making as much ATP again. Okay, because those protons are not going through the apparatus that can make the ATP. And so then DMP then short circuits this, or it actually leads to the need to consume more glucose. Right? So you are no longer, even though oxygen is present, you're no longer getting 30 to 32 ATP per glucose molecule. You're getting more like, who knows, it depends on the concentration of DMP, but it could drop all the way down to 5 ATP per glucose molecule or something like that. It's just the number I picked. And so there'd have to be more glucose consumed to meet energy demands. Okay? Right. So that's super, super awesome stuff. Okay. Now let's go back here. Okay. Yeah, we've already talked about my red check minus. Will grades be fully updated before the test? So I've already dealt with that. So at the very beginning, practice short answer or practice exam short answer. I got that. Curve for the class. Difficult to say at this point in time. I will say this: historically, no, I have never done that. I can say this: we have quite a bit of extra credit, more extra credit this semester than I've ever given. So I think that's going to affect grades, and I don't anticipate needing a curve based off of the results that I've seen from the exams and where we stand right now. Could be wrong, but anyway. Okay. Do we need to know how to calculate PO ratios for the exam? Yeah, I would. Yes. Yeah, that's the most straightforward answer. Yes, I would definitely expect that you be able to predict a PO ratio for NADH or FADH2 based off of the amount of subunits in the C ring. Okay, for sure. Okay. Okay, let's see here. Electron transport chain coupling. Okay. Let's see here. How do inhibitions of complexes affect ETC? Okay, so let's deal with this electron transport chain coupling. And then we'll move to some of these other questions. So electron transport chain coupling is kind of seen here, but let me go ahead and redraw this, okay? And I'm going to get even more detailed here in my drawing, and we'll put all the complexes in this drawing, okay? Because I think I'll be able to use this for some of the other questions I saw popping up too. So we're going to put complex 1 here, and we'll put complex 2 here. We're certainly not going to be drawing high resolution structures of these, but we're going to get some idea of them, okay? Complex 3 will be here, okay? Complex 4, of course. And then finally, ATP synthase. Still not a great drawing, but better than what I've seen. better than what I've done before. Okay. So now I'm going to put the membrane in here. And apparently the membrane is going to go up. Okay, that's it. Here we go. Complex. Sometimes this is called complex 5, or ATP synthase is another name for it as well. Okay. All right. There we go. Okay. So now we're going to put here P side, and we're going to put here inside. Okay. And so we know this. Hopefully you have and are grasping this concept that NADH is going to come in here, it's going to drop off electrons, 2 of them, okay, and those electrons are going to be passed to coenzyme Q, so I'll just draw QH2, and then it will transfer over here, okay, and it will transfer electrons to cytochrome C, that will carry them here, and those electrons will be transferred to oxygen, making water. Okay, and in that process, 4 protons will be pumped through this, 4 protons will be pumped through this. Of course, we got a lot more detailed in class than I'm being right now, but this will do in terms of answering the question in terms of coupling. 2 protons there. Okay, and of course there's this other process. It takes succinate, converts this fumarate. Okay, and those electrons end up on QH2 again, and those then are going to follow the same path that the other ones did. Okay, and these also will be pumped. Okay, now when we then look at this, okay, these protons are then going to move. So we got protons, all these, let's say 0.2 protons on this side of the membrane. Okay, and then these are going to move through here as discussed, go through this, come out here. It's going to allow for ATP production. Very cool. Okay, now when we think about then coupling, okay, because that was the context of the question here, coupling, what does that mean? Well, what it means is simply that these 2 processes, so if I were to draw a big red line in the middle of this, you have the process on the left, which is electron transport chain, and you have the process on the right, which is ATP synthase, okay, and ATP synthesis. When we talk about coupling, what we mean is those 2 processes, the one on the left and the one on the right, are codependent on one another, meaning this, that the process on the right can't run without the process on the left, and vice versa. The process on the left can't run without the process on the right. Okay, so the process on the right depends on the process on the left. I feel like I'm in a Goofy movie right now. Disney, some narrator of this. The process on the right, the process on the left. I'm sorry. So let's use different names. But the process on the right, which is ATP synthesis, depends on electron transport chain to provide the electrochemical gradient that provides the energy to make ATP. So it's coupled to electron transport chain because it can't make ATP without electrochemical gradient. Okay. Now, the process on the left, which is electron transport chain, it depends on ATP synthesis and synthase to move these protons, these guys, back across the membrane. Right? And if that's not there, then it can't, the electron transport chain can't run because the P side gets essentially quote unquote full of protons. It's the gradient becomes too steep. And so then it's coupled to ATP synthase because ATP synthase maintains a proper electrochemical gradient so that the energy found in the movement of electrons from NADH to oxygen is enough to keep pumping protons across based off of how steep or not steep the gradient is. Okay, so that's what we mean by coupling. Okay, of course, uncoupling means that DMP or something like it can independently move protons from P to N side. And when I say independently, I mean independently of ATP synthesis, the process on the right, and therefore can uncouple the electron transport chain from ATP synthesis. Okay, so that's what we mean when we say uncoupling. Okay, all right, so hopefully that deals with that question. Let me come back here and make sure that we don't have anything in the chat. Okay, so we do have something. Oh, I have several things. I'm sorry about your dog getting cancer. Yes, I'm sorry about that too. It's been very sad, kind of week for us. He's a great dog, so he's doing great though. He has no idea what's going on. So, but it's been sad. All right, but anyway, why is this thioester relevant in PDC? Let me come back to that because I'm still kind of in this thought process. I mean, Captain America would not be without a shield, especially in dark times such as COVID-19. So the vest, right? I know. Okay, I get it. The shield is the vest. I get it. I see what the connection you're making. But anyway, okay. So let's come here. Is ATP synthase still functional with the presence of DMP? That's right along this question that we're asking. And yes, it is. So unless there's some other inhibitor of ATP synthase, it can function in the presence of DMP. It's just not getting as many protons as it normally would. So the rate of it would actually and can actually go down. So anyway, but it would still be functional. Okay. Why is FADH2 drawn to move when it doesn't leave the enzyme? That's a great question because people have done it for so long and yet it doesn't leave the enzyme. So it's a great question. It actually, coming back to, so just to give all of you some idea of what's being asked here, FAD is right here, right? And it's going like this. I don't know if you hear the barking in the background, but that's Dickens. So he's enjoying life. He's outside barking at the dogs that are walking by the fence. So he's doing good right now. But anyway, so anyway, but FAD never actually leaves us. So the question is, well, why is that drawn? And I think it's just because, to try and represent the citric acid cycle in a cleanest fashion as possible, and that FADH2 is produced in that, it's often been drawn as this arrow coming out of that succinate dehydrogenase reaction, which gives the impression that it's leaving the citric acid cycle and going to the electron transport chain, when in reality it's not. So that's, I think, my best answer to that question. So, okay. All right. So let's come back here. Okay, there's no more questions. Okay, so why is the thioester relevant in the PDC? So that's actually coming back to this. We'll get back to the Q&A here in just a second. But let me come back here. This thioester. Let's look at this here. So thioester here. Okay, and there's another thioester here. Okay, and when you hear the word thioester, you should think this is a great way to store energy. It's a high free energy arrangement of electrons. Okay, kind of like when you think of Captain America, you think of first a shield and secondly me, and then apparently third a vest. But anyway, so thioester is equated with high free energy. Okay, so they're great ways. This bond is a great way to store energy. Okay, and the redox reactions shown here, and if you go back to your notes and look at this, I didn't really talk about it when we talked about the arm, but there's a redox reaction that's occurring here at this second step. It's really high energy redox step. And that energy, instead of being released as heat, is stored as a thioester, okay, between the acetyl group and the arm. And then the arm just passes that acetyl group onto coenzyme A, and coenzyme A has this thiol group in it that can reform the thioester. So the energy found in this process, okay, in particular that redox reaction, is now stored in this thioester, okay, in acetyl-CoA. We could map this same thing onto alpha-ketoglutarate dehydrogenase complex, and we could look at succinyl-CoA. It would look very similar to acetyl-CoA, but it would have more carbons, but it would have a thioester as well. And that thioester stores free energy from, again, the redox reaction. And in that case, we see that energy is used to make the ATP or GTP in the citric acid cycle in the substrate level phosphorylation event. So, okay, so that's why that's relevant. Okay, good question. All right, so let's come back here now. How do inhibitions of each complex affect the electron transport chain? So let me answer that. Let me answer that in as much detail as possible, while at the same time being general enough to help you problem solve this, because I can't possibly address all possible inhibitions of the electron transport chain. But let's just do this, okay? And this is why I wanted to draw this in this detail, because I saw that question. And let me go ahead and remove this red line, because I'm going to put another red line in this system, okay? So we've got these arrows, purple or blue arrows, that are representing how electrons can move through the electron transport chain, okay? And inhibition of the electron transport chain stops movement of those electrons represented by these arrows. So let's, for instance, say this, okay? And if you just kind of conceptualize the electron transport chain in this way, if I were to put a big red line right here, and that big red line represents inhibition of the processes in complex 3, then you start to see how electrons will stop moving through the electron transport chain, okay? They will stop right at this complex 3. Okay, now I have described it this way, and I can't remember if I used this analogy in the fall or not, but I'm going to go ahead and use it again, even if I did use it in the fall. So if you've heard me use this, then this will be kind of old for you, but I'm going to use it again. So if you think about the electron transport chain, complex 1, 2, 3, and 4, as essentially a room, if you will, or even like a hotel lobby is how I've described it in the past. And that hotel lobby has 2 front doors. It's got a front door here, and it's got a front door here, okay? And electrons, which would represent or people, let's say, which would represent electrons, can enter into the hotel lobby through complex 1 or 2, okay? And then they can walk through the hotel lobby, and then it has a single back door, and that single back door is going to be this, oxygen, okay? And then the people can leave the lobby through the back door, right, as long as oxygen's present. Okay, so people will enter in through the 2 front doors and leave through the back door. Okay, now this red line then that I put up, this inhibition, represents a partition that can occur in the hotel lobby that will be slid into the middle of the room, and all the people or electrons on this side of the partition, the right side of the partition, will leave through the open back door. Oxygen's still there, and all of those redox centers will be, of course, oxidize. They'll obtain an oxidized state because the electrons or the people have left. The room is empty at that point. The front doors remained open though, and so people or electrons kept coming in, but then they ran into this partition. Okay, and when they ran into this partition, they started to back up until finally all of that room was full, and essentially all of those redox centers obtained their reduced state. Okay, all right. So anyway, so that's inhibition. Now you could move that partition and say, oh, well, complex 1, will be inhibited, or complex 2 will be inhibited, or complex 4 will be inhibited. And you just move that, and you have to kind of then think about, okay, well, if I move that partition, what's going to happen to the electrons? Okay. Now, in this process, let's do this, and this is the second part of the thing that we really need to think about here is this. Once the hotel lobby gets full, okay, NADH, for instance, will stop going through the front door, meaning it will not be recycled back to NAD+. Well, at the same time, you have in the electron transport chain, I'm sorry, not in the electron transport chain, but in the mitochondria, you have this nifty-difty cycle going on. Okay, don't ask me what nifty-difty is, but it's an expression, I guess. Okay. Citrate is made, of course. Come back here, right? And even this, we could even do this. Pyruvate, we know, is converted to acetyl-CoA. Well, this NADH, can come and it will essentially inhibit these steps as it starts to accumulate. Okay, so those will start to then, the citric acid cycle and the production of acetyl-CoA will start to be reduced. The same is true of beta oxidation. So we haven't really mentioned beta oxidation in our review yet, but beta oxidation, of course, it takes the acyl-CoA, okay, and makes this. And of course, a bunch of NADH and FADH2. This will come back, and this will inhibit that, okay, as well, because just by feedback inhibition or like competitive inhibition, that kind of thing, okay. And so the whole thing will kind of grind to a halt in the mitochondria, okay. And then, of course, on the external part of the mitochondria, the cytosol, then the cell is going to detect that there is not as much energy production taking place in the mitochondria, and so then there's going to be an increased flux through ATP-producing pathways in the cytosol, glycolysis being the key there, and you're going to consume a lot of glucose. So that would be true of inhibition, or that would be true of just removal of oxygen. You're right. I mean, that would be putting the partition essentially right here is just closing the back door. If you remove the oxygen, the cell goes anaerobic, and that whole thing, even beta oxidation, shuts down. So, okay, so that's how inhibition works. Okay, all right. Let me go here and see if there's any remainder questions on this. Okay, so does ATP synthase rate increase or decrease when the presence of the uncoupler? Also, an interesting question. It would, in more than likely, decrease because it's not getting as many protons from the electron transport chain, right? The electron transport chain is it's pumping protons, of course, to the p side, but those protons are being stolen, if you will, from the uncoupler, and so its rate would actually go down because it's just not getting the protons. Now, simultaneously, the cell would detect that ATP is not being generated as quickly. And so then you would have increased rates or flux through pathways that can carry out substrate level phosphorylation. Specifically, you would get increased rates through glycolysis. You could even get increased rates through beta oxidation and even citric acid cycle, because we haven't removed oxygen; it's still there, so you can still use NADH. It's just that the protons are not being used by ATP synthase, so its rate will go down. Okay, good question, interesting. Okay. Okay. So we've answered those. I think we'll get about. Let's say we have about 10 minutes left. Okay. So let's move on here and see if we can't get through a couple more of those. What determines the amount of subunits in the C ring? If 9 is ideal, how do we get that? That's a great question that I honestly I'm gonna plead the fifth on this, or just essentially say I don't know what determines that. I've never investigated it enough to know. I don't know. Honestly, this is me speculating. I don't know honestly if it's a tightly regulated process or if it's just an assembly process, meaning that it's some form of random assembly that, the C subunits are put in there and they're just sort of random. I tend to think that it would be regulated. That would be my gut instinct on this, considering biology and the way things are regulated. How is it regulated? I don't know. So it would have to do with genetic expression of the C subunit and how much is actually being expressed. And we'll just leave it from there because I don't really have much more to say about it. So, interesting question. I just don't know the answer. So, and quite honestly, maybe the field doesn't know the answer. So, okay. Okay. Can you explain lipoid arm? We've done that. Do we need to know citric acid cycle and beta oxidation as in depth as glycolysis and gluconeogenesis? So again, start with diagrams. Make sure that you can connect all the diagrams and all the different pathways from the diagrams and understand that big picture first so that you can start to make sense of the details. And that would include, of course, putting the citric acid cycle and the beta oxidation in there. Okay. So again, I don't discourage memorization, but you're not going to be rewarded for it. Okay. Beta oxidation is a significantly smaller pathway than, of course, glycolysis. And honestly, you could remember it this way. It's very simple to remember what happens. You can remember it as being, it starts with a oxidation, it goes to hydration, oxidation, and then cleavage step. That's the 4 steps of beta oxidation. Oxidation, hydration, oxidation, and then cleavage, making the acetyl-CoA. Okay, and of course the 2 oxidation steps make the either FADH2 in the case of the first step, or NADH2 or NADH in the case of the third step. Okay, so anyway, so you're going to need to know the details of those to understand the language of the exams for sure. So okay, all right. Do we need to know? Have I already addressed that? Insulin glucagon with malonyl-CoA inhibition, right? Okay, so let's deal with that. That's a good one. So interestingly, I've already answered this question. Can you explain what happens to citric acid cycle and anaerobic metabolism? So we've addressed that. It gets shut down. Okay, we addressed that right here actually. So okay, so let's look at malonyl-CoA inhibition and insulin signaling and all that wonderful jazz. What is a good way to diagram this for you all? Okay, let's do this. Let's start with Cat1. Okay, so Cat1 is part of that carnitine shuttle, and you say, oh, a shuttle, I hate shuttles. Yeah, and they're just part of biology. Okay, so, but Cat1 is a part of the shuttle, and what it's going to do is it's going to convert an acetyl-CoA to acylcarnitine. Okay, and then when acylcarnitine is made, let's go ahead and draw this here. Okay, that acylcarnitine can make it into the matrix okay, while at the same time a carnitine is shipped out. not an acylcarnitine. So the acyl refers to a fatty acid chain. Okay, that's what we refer to. And then carnitine is just a small molecule attached to that. Oops, I spelled it. Too many N's. Carnitine. Okay, and this is, we've got another membrane here, like this. Okay, all right, so that's that. And then once we get the acylcarnitine over here, I'm kind of running out of room, but once we get that, then it will go through beta oxidation. So we can just go like this, 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 and say beta oxidation occurs. Okay. The key regulatory step of beta oxidation is getting the production of acylcarnitine. Once acylcarnitine is made, it will be moved into the matrix of mitochondria, and beta oxidation will occur. Okay. All right. So this is, quite honestly, even though it's not part of the rate of beta oxidation, this Cat1, the production of acylcarnitine, is the rate limiting step for beta oxidation. If you inhibit it, then you inhibit beta oxidation. All right, so let's then go to malonyl-CoA. Okay, it turns out that malonyl-CoA does indeed inhibit Cat1 at high enough concentration, and therefore inhibiting beta oxidation. Okay, well, malonyl-CoA is made from acetyl-CoA. Okay, acetyl-CoA is this 2-carbon molecule with a CoA enzyme A attached to it. This actually is going to be a 3-carbon molecule. Okay, malonyl-CoA is you add a carbon to acetyl-CoA and you make malonyl-CoA. Okay, and the enzyme that does this is called ACC, or acetyl-CoA carboxylase. This, incidentally, is the very first step in fatty acid synthesis. Okay, so we could then do this. Malonyl-CoA not only does it inhibit Cat1, but it will go on and be used with lots of other things, and so we'll just draw lots of arrows here. It will be used to make fatty acids. So that's the very first step in fatty acid synthesis. Okay? All right, so ACC activates this process. Now, ACC can exist in the form that you see it here, or it can exist in this form, ACC, okay, phosphorylated. Okay, and I'm going to put sunbeams coming out of this. Remember, sunbeams are universal sign for active. And then I'm going to put a little minus sign in this. When it's phosphorylated, it's inactive. Okay, well, to dephosphorylate it, this process here, we're just going to say this, insulin signaling causes the dephosphorylation. It activates this protein called PP2A that will dephosphorylate it, okay? And that will make the ACC active, that will make malonyl-CoA, and then that malonyl-CoA will inhibit Cat1, okay? This process, glucagon or epinephrine signaling or even low energy charge in a cell can cause that phosphorylation. It will activate pKa, and pKa will then phosphorylate it. Okay, so now let's try and understand this. Okay, from this conceptual level, insulin is secreted by the pancreas during times of a fed state. So if you eat something, your blood sugar goes up, insulin is secreted. Insulin then tells the liver to absorb glucose and also to make lipids. Okay, so if you have excess glucose in your diet, we know, okay, how glucose can be converted to pyruvate to acetyl-CoA, and then we can see here, once you get to acetyl-CoA can be used to make fatty acids, and the signal from the bloodstream is, yeah, make fatty acids, okay, and at the same time inhibit beta oxidation. So you're going to make fatty acids and inhibit beta oxidation. Okay, all right, and then glucagon, of course, says, oh, syn sugar, okay, and we have a low energy environment, and in that case, malonyl-CoA production will go down, You will not see as great of inhibition of beta oxidation because the liver cell will start to use lipids for its own energy needs and will start to make glucose. Okay. All right. So hopefully that answers that question. Let's go here. Come back to here. Okay. All right. So let's deal with this question, and then I'll go back to and see if we can't do two more. But let's look here. Why does NADH pump 10 protons and FADH pump 6 regarding PO ratios? Okay, real quickly here, let's do this. Come back here, okay? So when we start to think about 10 protons pumped by NADH or 6 protons pumped by FADH2, let me draw this. NADH here, okay, is going to lead to, I'm going to circle these, 4 protons pumped through complex 1. Those electrons make it onto QH2. Those will go through complex 3. There's another 4, so that's 8. And then those electrons will get onto cytochrome C and come to complex 4 and pump 2 more. So that's 4 plus 4 is 8 plus 2 is 10. Okay. The FADH2, which really sucks in, it gives us electrons. Those electrons go through complex 2. They bypass complex 1. And those then go into complex 3 and pump 4 and 2. Okay. 4 from complex 3, 2 from complex 4. That makes 8 equals 6. Okay. So hopefully that makes sense to you. If not, go ahead and contact me and I'll try and explain it further. Okay. All right. Okay, curve for the class, we already talked about that. Explanation of TPP, lipid, and honestly, I don't have time to answer that question. So, okay, I think that is the best one. We've already talked about that. We already talked about a short answer. Okay, so do PO ratios, let's see if this is a quick one. Do PO ratios include the impact of extra hydrogen needed to bring phosphates back into the mitochondria? E.g., a 9-unit rotor consumes 3 hydrogens to rotate 120 degrees and bring one to bring in the PI. So let me see here. Do PO ratios include the impact of extra hydrogen? Yes. Okay, so I understand what you're saying. Yes. So when you think about PO ratios, you do have to consider how many protons are being used by the subunits of the C ring to make the ATP plus the 1. You always add 1 to bring inorganic phosphate into the mitochondria. So yes, they do. Okay. Let me come back here. Explanation of TPP, lipid, and FAD, part of the PDH complex. So honestly, I think that I would reference you back to this. Okay. Let me come back here. So I'm going to reference you to this slide, which we spent actually quite a bit of time thinking about this, even today. Okay. TPP is simply just decarboxylating pyruvate. Okay, we talked about the lipid. Okay, so TPP is just doing the first step in this. Okay, okay. So I know that's real brief, but I do have to go. So, okay, so let's go ahead and stop there. Oh, and FAD. Let me. I guess we'll just come back here. FAD, right? Is going to accept electrons from lipid. And then FAD will give electrons to NAD plus, making NADH. Okay, so step 4 is get electrons from lipid. Step 5 is give those electrons to NAD plus, making NADH. Okay, okay. So I think that's a pretty good place to stop right there. Okay, this is one of the last live interactions I'm going to have with you. I've had a great semester. It's been a strange semester, but it's been a good semester in many ways. So please take care of yourself over this next couple of days. I hope that every single one of you do really well on this upcoming exam. If you have any questions, go ahead and email me those questions or contact me in some way, and I will hopefully be able to get back to you. So, okay. So, and in the future, right, if you need something from me, let me know. If you have some awesome biochemistry question, let me know. I'm more than willing to help students, even if you're not in my class. So, okay, I'm here to help you guys. Okay, all right, take care. I will see you all virtually, I guess, and of course on Monday. So, okay, all right, good luck on the exam.